Okay, so my great pleasure to introduce today's speaker, Dr. Rafe Brown. Rafe is the curator-in-charge of herpetology at the University of Kansas Biodiversity Institute. That's their natural history museum. One of the most important and famous herpetology programs in the country, with a huge collection that Rafe oversees and has spent a lot of time contributing to. Rafe did his, just by way of his academic background, he did his undergraduate degree at Miami University in Ohio. And then he followed that up with his master's work there. He was working with Sheldon Gutman there on the phylogenetics of the Rana complex. And that's when he really had initiated his work in the Philippines. And Rafe, of course, is super famous for all the work that he's done in the Philippines. Rafe then went to the University of Texas at Austin and was a PhD student there in the labs of David Canatella and David Hillis. I was in the same lab. Rafe showed up there a few years after I started there. And actually first met Rafe when we were doing we met in the Philippines, and we did a bunch of field work together in the Philippines. We did field work together in Indonesia. So we go way back and have done a lot of collaborative research over the years together as well. Rafe is one of the most productive scientists you're likely to encounter, actually. So he started a few years after I did at UT Austin, but he's already basically lapped me in publications. Rafe has more than 275 publications, huge H index, super productive. By this time next year, he'll probably be over 300 publications. And he has a very Dave Wake-esque like research program where he does everything from describing species diversity in the Philippines, and he's described. Do you know how many species he described? Dozens, right? Like maybe hundreds even. Yeah, so he's described tons of species like Dave Wake would do, but he also writes many highly conceptual papers published in the very top journals. And the work we're going to hear about today is going to include some of that research. So very lucky to have Rafe here. And Rafe, the floor is yours.
Thanks, you all. It's so great to be back. I was here for a postdoc in 2004, 3, 4, and 5-ish, right around in there, and one of those years, and hadn't been back in almost a decade, and it's so cool to be back and be in person. This meeting is being recorded. Got it. Okay. So yeah, thank you very much for the invitation. It's a real pleasure to be back. I really had fun at Herp Group, and what a pleasure. I get to talk twice. How cool is that? Cool. Today I'm going to talk about the Philippines. So, okay, audience participation. Who's been to the Philippines? All right, good deal. About 25% of the room. Good. No, I'm just saying. It's a great place. I strongly recommend going to the Philippines, and now it's more fun and safer and more accessible than ever. It's a great place to be a tourist, go on vacation. Filipinos are fantastic. I know, we love Thai food and Indonesian food, and Filipino has its own tradition, but I actually really love Philippine food and crave it when I'm not there. So I strongly recommend people going to the Philippines if you ever get a chance. The Philippines has this amazing, iconic land and seascape, island archipelago, volcanic island archipelago landscape. And if you get to travel around the Philippines and see all these different shapes and kinds of different landscapes and the biodiversity there, you'll never forget it. It's really quite an experience. And so right in this picture, for example, on the Bicol Peninsula, which is just a long peninsula that's just volcano after volcano after volcano. It's striking, right? It's right on the edge of the Ring of Fire. It's a beautiful place, and it's packed with biodiversity. And so what's the big deal about biodiversity in the Philippines? It's more than, this is a landmass the size of only Arizona, if you put it all together, the US state of Arizona. The whole Philippines could easily fit right inside of California, but it's more than 7,100 islands. And the Filipinos will even, if you ask a Filipino how many islands are in the Philippines, they'll ask you mean high tide or low tide, right? I mean, they are acutely aware of the diversity of landmass there. But we know that it was about originally 95% forested, and that's down to about, 4 to 6% of the original forest existing today, but lots of secondary forest coming back and lots of consideration of biodiversity and other types of habitats. It has the distinction of only with Madagascar of being one of the planet's both megadiverse countries and global conservation biodiversity hotspots. And that distinction puts it in this category along with, Madagascar, right? So it's that kind of a place. The diversity is incredible with over 50,000 described species, half of which are endemic, and more than 1,000 endemic land vertebrate species. These are 4 species that were described as part of our research program. the Scooby-Doo mystery machine frog, a new species of bird described in 2014, a new genetic type of tarsier. I mean, I'm a herpetologist and I get to work on the genetics of primates, right? Really cool. A giant 2-meter long, entirely frugivorous monitor lizard that we discovered in 2010 that scientists hadn't seen, but of course the indigenous communities that were there who showed us this animal for the first time, they had seen it and they had a name for it. And so when we named it to science, we chose their name. Incredible vertebrate biodiversity. So how is it that scientists have gone through the process of trying to understand that biodiversity and put it in context? So much endemic land vertebrate biodiversity packed in such a small amount of space. Well, the development of the Pleistocene aggregate island complex paradigm has been sort of the way in which for the last half century or even longer, 3/4 of a century, people have been trying to understand biodiversity in the Philippines. And a lot of that goes back to the work of Edward H. Taylor, who was a KU professor who went to the Philippines in the early 1912 through 1924, lived in there in between, spent time in that bracket of years there, living in the Philippines at one point for about a year and a half, describing a lot of the diversity, publishing papers on the lizards, amphibians, snakes, and turtles of the Philippines. And he published this wonderful paper in 2028 in the compendium by Roy Dickerman was an ornithologist, right? Dickerson, about distribution of life in the Philippines. And Dickerson he was at the Bureau of Science, compiled all these information about different groups of organisms in the Philippines, including Merrill's work on botany and plants of the Philippines and forest types and invertebrates, and put it all together in this place. And Taylor had a chapter where he recognized these 5 different faunal regions of the Philippines just based on species diversity. He didn't know anything about the geology, and he didn't know anything about the way we would define, different land masses or shelves or the underlying kinds of rock and dirt or anything in those environments. He just was recognizing them on the basis of what species were there, and most of which he had named, basically, or had to reconsider from this earlier period of exploration from European early collectors and when people first sort of started traveling around the world and describing biodiversity. But he recognized 5 different faunal regions in the Philippines. And in this picture in his monograph, he's wrapping distributions of species around the island groups, some of which only go this far into this part of the Philippines, and some of which extend over this landmass here, which is Mindanao, and this last landmass Palawan, and they were shared across this distribution, and others in these sort of layered hierarchical waves of distributions that set the stage for this amazing work by Robert Inger in 1954. So I guess this is really like the late 40s that he was putting this together. And right there on page 455 of his monograph this is a taxonomic monograph where he's mostly describing species using telegraphic style writing, and one little chapter on biogeography and geographical trends. And there it was, the Pleistocene aggregate island complex, where he used channel depth sounding information from where ships would go around and fire charges at the seafloor and measure the time the echo took to come back to measure the depth of channels. They got information about the depths of channels between all these islands, and he inferred, well, that during the Pleistocene, when water was tied up in the polar ice caps and sea level had dropped about 100 to 125 meters, this is what the land masses should have looked like. So he laid out those 4 or 5 major geographical or faunal regions in the Philippines and the underlying geological platforms that formed those centers of biodiversity, those centers of endemism. Of course, at that time, Ernst Mayer's work and all the work on speciation theory was coming along, and people recognized this as the effect of isolation. These are islands that have permanently been surrounded by deep water channels that have never been connected to other land masses. And then there were islands that were connected, like Catanduanes and Wheeler, that we know that were connected to nearby Luzon. So that presence or absence, or that isolation or the presence or absence of land bridges, really, fit into speciation theory and the idea about peripheral isolates model of speciation. And it sort of all fell into place with this idea that if we knew something about channel depths and the 120-meter underwater bathymetric contour, we could infer which land masses were connected to each other and put together this idea of not 7,100 islands, but maybe 5 big islands that have their own history of isolation. And that's why they have their own faunas. And the faunas really made sense for not just reptiles and amphibians, but also mammals and birds and insects and lots of other stuff. So this near perfect definition of the Pleistocene aggregate island landmass systems took form. And of course, the secret to understanding that is that 120-meter bathymetric contour, right, traced around the land masses of today. This is underwater bathymetry here. and inferring what those Pleistocene seashores, where they might have been and what they might have looked like this big land positive peninsula that encompassed those two islands of Clelio and Catanduanes. It connected these islands, but never did connect to this island, Mindoro. So permanent isolation and periodic isolation and oscillating sea levels during the Pleistocene, right? We know during the mid to the late Pleistocene that sea levels fluctuated with the ice ages 10 or 12 different times. So where does that take us? Well, that has become this sort of textbook example. In fact, it's cited in the textbook that I used to teach biogeography. And here's some mammalogists who's made a lot of Robert Inger's observations of where land masses should have been. Exposure of the Pleistocene land bridges has had a profound impact on today's patterns of species biodiversity, much more so than any traditional biogeographic boundary or biogeographic variable. So, right on the heels of MacArthur and Wilson's equilibrium biogeography, this idea of land connectivity versus non-connectivity having this profound effect. And then eventually the extent of land and sea were formalized in the year 2000 by Harold Vorris's work when GIS technology became widely available and people defined those on a much finer scale and looked at the differences between 110 meters below sea level and 120s. And the idea of the distribution of land and sea became explicit and quantitative and geographical. But the Pike diversification model and this idea that the Pleistocene aggregate island complex system became the 50-year and now a really 75-year paradigm ever since Inger, which has been invoked over and over again to explain Philippine megadiversity and endemism and concentrated in different pockets of the archipelago. And if you do the calculations of the numbers of land vertebrates that are endemic to the archipelago per unit area, it looks like the Philippines has the highest concentration of endemic land vertebrate biodiversity of anywhere on Earth. And so that is very of general interest, conceptual interest to evolutionary biologists when asking this question: can these unrelated lineages have a common evolutionary history that is governed by this external system of oscillating sea levels and expanding and fracturing land masses, right? Islands that become bigger and then become smaller and fragment apart and become two separate islands, and then the sea level drops and they become one big island again, and then the sea level comes up like filling up a bathtub, and now you have two separate islands. Could that oscillation of sea levels result in a species pump mechanism where with lots of oscillations, more and more species will be concentrated as a result of that ephemeral isolation? But at some point, species might accumulate enough difference or become reproductively isolated as a function of all the different mechanisms that we all know about, and some of those species won't reticulate when islands come back together, but may persist even in the presence of land connection between them. So it has become this model for partitioning, for describing how the biodiversity in the Philippines is partitioned, how it's been maintained through time, and possibly even contributing to how it was originally generated. So that's what we're going to focus on a little bit today. So today I want to talk about that species pump mechanism and what are the expectations and what are the predictions and how can we go about testing them? So here's the idea, right? Here's a species pump system. The secret here is focusing on the difference between the permanent marine carriers that foster isolation and deep allopatric divergence between these taxa. So here are permanent marine channels, and this is the 120-meter oscillation of sea levels and the exposure of land and sea. Here's three islands, Luzon, Mindoro, Palawan, that remain permanently isolated from each other. And so we would expect them, just as Taylor observed, and Inger did with amphibians as well, to have three distinct faunas and lots of endemic species that only occur on Mindoro and differ from the ones that are only occurring on Luzon, et cetera. Pretty straightforward. On the other side, within the Pikes, within the Pleistocene aggregate island complexes, watch what happens here. Here's one big landmass, think about 10,000 years ago. Sea levels start to come up as the Earth warms, and it fractures or fragments into 4 or 5 separate islands, which then reconnected, et cetera, every 10,000 years or so, maybe 10 or 12 times during the Pleistocene. So we focused on the isolation. Of course, under species theory, that isolation is the secret to the accumulation of enough reproductive isolation so that when taxa come back together, maybe if there's dispersal across these permanent channels or something, when organisms come back together, maybe they can't interbreed and they become reproductively isolated. But here, within a Pike, within a Pleistocene aggregate island complex, is it possible that this sea level vicariance, so what we're looking at here is rising sea level vicariance and the fragmenting of that landmass, can that that vicariance, sea level rising vicariance in shallow time, over the end of the Pleistocene, could that have resulted in causing divergence between taxa? And can the pike paradigm not just describe how biodiversity is partitioned archipelago-wide, but maybe even how some species were first, where they came from, what their origin was as a result of recent sea level vicariance? And has that resulted in a permanent speciation or signals of species that we can find empirical evidence for today? Well, we tested this on lots of these similar predictions between like 2005 and 2013, and I won't bore you with a lot of those sort of, single locus genetic studies and phylogeographic context of back then. But we wrote this review article in 2013 and didn't find a whole lot of evidence using 5 or 6 different major predictions of the pike paradigm for this idea that the rising and falling sea levels generated biodiversity, contributed to sea level vicariance and speciation. We didn't find a lot of evidence, and so we sort of didn't want to completely dismiss the idea. And so we had a section of this on the augmentation of the pike paradigm and what we might be able to tell from the future. And we found lots and lots of exceptions to the pike predictions, very straightforward, traditional population genetic predictions and the analyses that we had available to us at those times. But we could clearly see, even though the daunting task was ahead of us here, that multi-taxon comparative studies and genomic data were the wave of the future. And we really wanted to focus on this idea that could sea level vicariance actually generate terrestrial biodiversity? And so that led us to codiversification studies, which I was just learning about here as a postdoc when MS Bayes and all those studies and the ranges of codiversification studies first were being developed. And a lot of that happened here at Berkeley. It was an exciting time to come from here for my postdoc to be thinking about this stuff. So here's an example of a review paper that we wrote at the end of this single locus phylogeography phase, where we went through all of our data, and I had mammalogists and herpetologists in my lab at the time. And we just identified pairs of populations where we had population level sampling and population genetic data. So for this comparison within this pike, within the West Visine pike, Panay and Negros, right? It was this channel that formed 8 or 10 times as the sea levels fluctuated in the Pleistocene. Did that generate any speciation that seems to be sister species that are split across those land masses? And we looked at this time through all the data that we had, including multiple groups of frogs, multiple groups of squamates, lizards, and snakes, all these different mammals, including bats, shrews, rodents, all sorts of things, just to see if we could find any evidence for sea level vicariance that would have generated speciation or diversification just across those land masses, and with all the caveats that come with the conclusions that you would make from it. And we'll go through all of those. But we published this paper in 2013 where we investigated and looked at that, because across all those populations, and here I just showed you, I think, 9 comparisons. No, this is about 12 or something for these different taxa. Across eventually what we packed into this paper were 22 different comparisons of group sisters, so what looked like sister species or putative species or populations split up within pikes. We found, using the method that was available at the time, very strong, posterior density support for, this is divergence time in generation versus variance in time. Very strong, what looked like evidence for this idea of a single divergence. And if we assumed a 1-year generation time and a 2% molecular clock, which comes with all of its assumptions and caveats, it looked like, 18,000 years ago, right at the onset of the last, glaciation or warming period, might be the time where all these populations, all of them, diverged at the same time as a function of this non-shared mechanism, right? A shared mechanism of diversification across all these different taxa. That would be really exciting if we had strong evidence for that. But what we ended up doing in this study, and this is the work of Jamie Oakes, who was a PhD student in my lab and is now a professor at Auburn, associated with this power analysis of these data, we basically arrived at the conclusion that the available methods at times were positively or biased towards that conclusion of clustered events, which we saw as a potential weakness of the method if people were using it in a naive way, and concluded that the interesting, sexy finding, like the one we just found, 22 different vertebrate groups, putative sister species pairs, all diverging as a result of the same, external mechanism, if people were biased towards jumping to that conclusion, the conclusion that would get them, the publication or the the exciting result that they were most excited about finding would be co-diversification. And so Jamie went on to find other ways to implement the method, and eventually, among those 22 populations, we found all these different possible implementation, less support for highly clustered shared temporal divergences, and uncertainty in the number of divergence times across those 22 pairs. And actually, at this point, we became much more comfortable with these methods when we could. I'll take the variation rather than the conclusion that that you might rush to publish on and then be terribly wrong about, right? So full Bayesian computation was an improvement over the approximate Bayesian computation era in that method, and that was all Jamie's work. But let's go on and talk about more recent work with Philippine geckos. And this was geckos in the form of the genus gecko and the genus Ceraudactylus were these groups that we had sampling from throughout the archipelago. And because those were ones where we just had a lot of sampling, they became the ones that we focused on, even though geckos in a lot of ways could find ways because they are squamates and they disperse over water barriers really easily and can move around on floating debris and all sorts of other things, have specializations like eggs that are resistant to saltwater, desiccation and things. They might be things that could be exceptions to the strict terrestrial environment constraint, but in any case, these were the ones where we had great sampling because they're easy to catch. We had them from all over the archipelago. And gecko, the genus gecko proper, is more than 60 species across Southeast Asia. And at that time, there were about 14 endemic species limited to just the Philippines, and there's more now. And of course, they're nocturnal scansorial lizards that eat a lot of terrestrial vertebrates. They all sort of shared things in common. They had subdigital lamellae on the bottom of their feet for grasping vegetation and things and climbing. And what we had just published a paper in 2012 where we inferred that the origin of this whole clade was potentially a continental fragment, the Palawan microcontinent block, which ended up in this position. But originally, before the Western Philippine Sea rifted apart, was connected to the Asian mainland, and there was this period between 40 or 35 million years of it potentially existing as a separate landmass isolated in the middle of that rift valley that was opening up and becoming what is otherwise known as the South China Sea, before it docked into this position at 10 million years ago with the rest of the Philippines grinding up against it and allowed for dispersal of geckos into and throughout the rest of the archipelago. And so that was just a paper that we published in 2012 that inferred the deep history of gecko relationships and how they got to the Philippines and resulted in an archipelago-wide radiation of all these spectacular endemic Philippine geckos that we had sampled throughout the archipelago. So we wanted to focus on more shallow time, early divergence, and to see whether the subsequent oscillating sea levels when the Philippines was in this configuration could have generated more of that species diversity than the one or two lineages that rode the Palawan raft to the Philippines. So that's gecko. And then the genus Ceraudactylus are the bent-toed geckos that have claws rather than subdigital lamellar pads. These are ones that they climb a lot of vegetation substrates in forests and have this very interesting, unique among gecko claw morphology. And we published a couple papers on their phylogenetic relationships and knew a bit about them. And some of them diverged before the time frame that was convenient for the Pleistocene, but a lot of them look like they might have been affected by this species pump mechanism. So we focused on 8 pairs of geckos, 8 pairs for both gecko and Ceraudactylus. And these are the ones we chose throughout the archipelago. And we inferred ones that were definitely connected by land masses like Panay Negros in blue, some that were maybe like Tablas and Sibuyan Island. We know that Tablas and Romblon were connected, but Sibuyan might have had a shallow, narrow ephemeral channel between it and these. So for big land masses where we knew there were broad land bridges, and then others where we inferred that Bohol was part of the Mindanao pike, but we don't know whether this island Camiguin Sur really was or not. So we don't know whether Bohol and Camiguin had dry land between them. So we had this sort of range of things that we knew were connected by broad land bridges, some that weren't, and some that may have been or were connected ephemerally. And we collected RAD-seq data from throughout these groups for about 400 individuals. So it's a big, huge, giant new genomic data set that we had to compare these 2 different clades that were co-distributed in the archipelago with big overlapping distributions, but had different original evolutionary histories. And so in this group, so this is a paper that we published in 2018, I think it was. 2018 was 2019 when this second paper came out. In this case, we found strong support for 8 pairs of separate pairs of species or putative species diverging independently with no evidence of diversification. So this is different. We're looking at the numbers of independent pairs diverging independently. And it looked like among those 8 groups of cerdactyls, the net-toed geckos, they were all separate. Right? There weren't co-divergences amongst any of those populations that made sense where we could line them up. There they are, right? These are the separate divergence times amongst those, and you can just eyeball them and see that none of them line up with each other. So across these different groups of things that we knew were dry land connections and ones that weren't, and things in between that may have been, we didn't find any evidence of co-divergences within the Cerdadactylus clade. Now within gecko, it was a little bit, there was evidence for possibly not 8 separate ones, but there may have been some co-diversification. And you can see one pair here in the data, two inferred posterior distributions of generation divergence date estimates here that overlapped. And so maybe 7 separate divergences, and maybe a couple of them lined up, but not much evidence, right? And we had like the best sampling, the best methods, these new methods that Tammy had developed, and we still weren't finding any evidence strong evidence for the pike paradigm generating, the species pump actually generating diversity with the evidence of temporally clustered divergences, which seemed like a pretty fundamental question. If these things were really, if speciation was being generated by this species pump mechanism, I would expect to see more than this. And yeah, of course, why would you expect to see things that have different generation times and different demography? And of course, all those assumptions are true, but these are 2 different great clades of closely related geckos. And this is about as closely related but separate as I was going to be able to get with this kind of sampling. And so I was really excited to find some evidence, and found very little in this paper. And we were sort of given up here, right? The lack of support for this primary prediction of the species pump hypothesis. Few, if any, temporally clustered divergences in the taxa. I was making up, pulling things out of my behind about what the alternatives were. Habitat heterogeneity, or rare, quote unquote, rare overwater dispersal. Well, probably it was not as rare as the vicarious biogeographers wanted us to think. And of course, we would expect some dispersal. So I was starting to think about telling that story about a dispersal dominated system. And here's a really cool paper that came out where rafting on vegetation and coconut is an experiment where some colleagues of mine, Hu Yang and Wen San from the museum in Taiwan, did an experiment where they rode the Kurashino current, which is this famous current that goes from the Philippines up through Taiwan and Orchid Island. And they actually put experimental rafts of coconuts and things out, and showed that for weevils, and they rode behind this thing and followed the semi-natural dispersal and actually showed that yes, coconuts and vegetation, mats of vegetation, were sufficient for dispersal of organisms between the Philippines and Taiwan. I thought that was really cool. Why had nobody done that already? Anyway, why are we arguing with the vicarious biogeographers when we could have just done that experiment? But anyway, species pump turned out to seem to not be the rule in Philippine geckonids, at least. And we had that with this replicated system. So check here, 41. Okay. A new generalized model, empirical results in geckos. So this is Jamie Oakes' brilliance here when he sort of had been thinking about this and what we assume about phylogenies. And we had this discussion, and he really developed all this and develops the credit for the thought process here. But all processes in diversification, the assumption in phylogenetics for these tests of co-diversification were that all processes in diversification impact lineages independently. And the idea is like whatever processes are driving the speciation, or the process of diversification of this green versus orange gecko here, should the assumption, ought to be that they are independent of and potentially different processes than what's driving this one here. Divergences, and another general assumption that ought to be the case, but it turns out it wasn't. Divergence, that it was the assumption, right? But what if there were situations where it wasn't, right? What if a common mechanism drove that actual divergence? And that's what we're trying to test. So we wanted to look carefully at those assumptions. Another one was that divergences only cause bifurcations, right? Splits between lineages. And the available models of approximate Bayesian computation and the way the tree was implemented in all these tests assumed strictly bifurcating process. And then, of course, the other one was that the divergence between bifurcating independently diverging species didn't affect other lineages. Whatever was ever going on affected those two lineages and didn't have a shared impact with other lineages. So Jamie implemented the relaxation of all these assumptions in this model. And we just basically want to consider something like this. In a case where there are three lineages, so here's three different groups of geckos. Think about the end of the last glacial cycle, 18,000 years ago, Earth is warming up, sea level's coming up. Some of these land masses, like this model over here on the right of the screen, glaciers melt, sea levels start to rise, and some of those models, some of those land masses start to break apart as sea level rises and they get cut off. Imagine two mountains on that island. And as sea levels come up, those two mountains become separate islands. And so for me, I'm thinking all the other groups of geckos in the Philippines, like Cerdidactylus gecko and pseudogecko. And during this time, I was thinking about this group, pseudogecko, that has another distribution of geckos throughout the archipelago. These beautiful forms that have a different kind of habitat. I won't go into all that, but that's what I was thinking was, okay, let's get more data, a third clade, test this with this relaxation of these assumptions. So back to our assumptions, imagine the single island, sea levels are coming up and it becomes, those mountaintops become new islands and now you have two islands. That would be a shared event where all these divergences or bifurcations should be affected similarly by the same mechanism, right? So this is, if sea levels rise and two islands fragment into smaller land masses, that causes all the lineages to diverge simultaneously, potentially, under an isolation speciation model. Clear violations of the assumptions of these independent divergences that were implemented in the available tests, right? And so let's just relax those assumptions. And so here's another way to think about it. The current methods only considered these trees, right? These bifurcating topologies that have strictly splitting into two lineages, bifurcating events, versus this one, where other trees that we might want to consider, hard polytomies, trifurcations, multifurcations, these are all different topologies that these models should consider. And until Jamie implemented them explicitly, they were violations of those basic assumptions and incorporated into these tests. So let's look back at our data here for Cerdidactylus. For gecko, we had 1,700 loci and about 1,000 loci, and you can see the numbers of the sites there. These are the populations from throughout the archipelago, slight expansion of the species of the sampling. And we're expecting that at the end of the Pleistocene, 18 to 12,000 years ago, in this case, the pikes are fully formed, sea levels rise again, these aggregate island complexes start to fragment and then become today's archipelago of separate land masses. Well, we'd expect aggregate island complexes to fragment. And if the separation of the pikes into today's smaller land masses impacted those resident lineages that were already there, we would expect, to see some levels of shared divergences. That just makes sense under that model. And as implemented, look what we found in this case. Strong statistical evidence for 6 shared divergences or multifurcations across Cerdidactylus. And you can see them here. This is in recent times for sure, but line them up across those clades. Strong, this is the first time, for me with the p-value to see strong statistical evidence for that prediction of clustered temporal estimates of divergences across this clade. And we had tried a lot of time, a lot in the past, and lo and behold, we found it. So for me, I was like, whoa, the pike paradigm is back. Woohoo, and it'd been 25 years. So, I was kind of excited about it, but all within the last 5 million years. So the temporal time frame for diversification is right. Here's gecko, the same thing, another 6 shared divergences across gecko, one of which was pretty old. Actually, some of these, there's here one at 5 million years and another one here at 10. So some earlier things before the oscillation of sea levels, but just finally some evidence and strong statistical evidence for shared divergences when we allow for things other than just strict bifurcations, which I thought was pretty exciting. So take home point with this method of biogeography, and there's a lot more to it. And I'm gonna give you some of the resources here if people are more interested in this stuff. I'm doing on time. Okay. This new approach implemented by Jamie Oakes, who's a professor at Auburn, and I'm happy to share the information that Jamie has decided to, after his 2-year sabbatical in industry, he's coming back to academia and gonna be back in place in academia here soon. So I hope that's okay for me to share that information. Yeah, it's great news. I'm really happy. Right? This new approach performs really well, and we can infer phylogenies with shared divergences quite well. It also performs well in simulations, even without the shared divergences, when people are trying to track other clustered historical events. which mean, analytically or computationally, it's no cost to the method. It's the first instance that I know of, that I've seen published, of strong statistical support for temporal predictions of the pike paradigm in recent shallow timeframes. The idea of cluster divergences across the Pleistocene, found in co-distributed clades, in unrelated organisms, and also within clades. So it has strong promise for multi-taxon comparative biogeography, and I'm really excited to run out and look at pseudogecko and a bunch of these other groups, and also focus on other groups that have different demography and life histories and generation times, because of course they shouldn't all, diverge just because the islands are changing shape. But, and then, I've been working in the Philippines for so long, I'm just really excited to see the Philippines at center stage for the development of new methods and pushing of the thought process forward and testing the predictions of ideas that have been around for more than a century, and these sort of textbook examples of biogeography that get cited uncritically, but we couldn't find any evidence for them, and then suddenly, when we asked in the right way, we did. So I want to talk a little bit about caveats and future directions and admixture, because the other side of that coin of the generating isolation, which is how we all think about how a lot of speciation gets started in some cases, at least in external, forces that generate species. The other side of that coin is when, these different land masses were connected, and we know that the whole was connected to Lake and Samar and Mindanao, and that there were dry land connections between all these things, between these land masses. The other side of that coin is, is whether the archipelago and the mechanism functions either as a species pump, but it also could just as easily be thought of as a species sink, right? Anything that starts to form in isolation could just as easily be wiped out at the next oscillation, right? And that admixture that you expect when the reconnection of those land masses go together, that's not something that people, in the biogeography literature really talked about for the last 50 years or 75 years. People focused on which of these land masses were isolated from each other and not so much, and then within them, which ones were fragmented, and that's why we find a subspecies on the whole that's different than the subspecies on Samar and Leyte, which is different than subspecies. I mean, people are just focused on differentiation, the taxonomy, how the differentiation might have come about, and not on the flip side of that coin, which would have been, what about taxa that could have been there and been very distinct and have been, reticulated back into what we find today as a single species. And there are lots of species that are found throughout this whole landmass, the pike landmass. That's very different than the within pike comparisons should generate a different set of predictions than the among pike comparisons. And we tend to focus on this isolation when we think about where species ought to form, and that has dominated Philippine biogeography and 5 separate faunas and throughout the archipelago. And maybe, although there is a very highly distinctive Mindanao faunal region and fauna, I wonder how many species were lost and whether that system has functioned more as a species sink than a species pump, right? Some of this work that we've done recently on Borneo has shown just astronomical levels of admixture across heterogeneous landscapes. And a lot of the taxonomy has proved to be wrong in some of the species group. This is work by Chankin on Borneo and the frogs of the genus Pulchrana. My master's work that Jim mentioned at the beginning involved a group that was related to them throughout the archipelago, and we found lots and lots of admixture within the groups in the archipelago. In fact, the really interesting things are highly endemic species that seem to have, where each sample within certain land masses seem to have the same proportion of ancestry coefficients or of genomic backgrounds across these groups that look like, the highly endemic micro sort of endemic range limited species tend to be ones that come out in our structure like plot with genomic data. This is 14,000 loci from frog-cap data. Turn out to be these ones that are phenotypically really distinct in some cases and not intermediate between 2 parental species, and seem to be a third taxon that's no taxonomist would ignore and name it as a separate species, and yet they sometimes look like the heavily admixed populations. But lots and lots of admixture across the archipelago between biogeographic zones. Like, look at what's going on here between the light blue of Samar and Leyte and the dark blue of Mindanao, right? This is a place that did have broad dry land connection between those land masses, and yet still there's all this admixture. And so the idea of the species sink and promotion of admixture and gene flow and movement across those land masses is something that's come up a lot, especially within our island archipelagos. We hear about this kind of research in continental systems where heavy, big, broad areas of sympatry between pure genomic things on the eastern versus the western side of North America, and then a big period of blending, you know, of contact between pure areas of pure ancestry. There's another group, but we don't hear so much about that in our island archipelagos. And so, you know, combined together with our expectation of isolation and speciation theory in island archipelagos, and now we find all this admixture, I'm just really intrigued about that being a fundamental component to the diversification process, and how admixtures and novel combinations of genetic backgrounds may promote diversification. Here's a study with 6,000 loci of puddle frogs from throughout the Philippines, and you see some generally looking, you know, in genomic background or genomic types in the, you can see the shading behind the pikes, or the green versus orange versus red, blue versus purple, you know, the geological platforms of the Philippines. And some of those genomic backgrounds and the genetic data correspond to that geography, but some of them don't. And there's a lot of interesting admixture at these sutures between paleo islands like Bicol Peninsula versus the rest of Luzon. You can see what's going on here in areas where types come together, and those populations that we've sampled are the most heavily admixed in these areas right here. This is also work by Chunkin on that was published in SysBio just recently. And I'm running out of time, so I'm going to let zoom forward to some resources here, but that's my general interest. I'm really interested in getting feedback. And so if people have ideas about admixture and novel genomic backgrounds and combinations and potentially hybrid speciation in island archipelagos, I'd really love to talk to you about it. I want to point you at some of the resources that Jamie's made available for his new method. The generalized tree distribution, the validation of his new algorithm, the performance tests, and all the simulation studies are available. You can take a look at Jamie's 2019 and 2021 presentations at Evolution meetings. The sea level animations that you saw earlier are available here. The software package, Ecology, and then the new FICO eval that we just talked about today is all available here. So we're recording, so I know you'll have those resources. And so, you know, my idea for future studies along this line of inquiry involve, you know, the continued focus on sea level vicariance, but we're interested in, as I said, these different groups of things that have different life history and demography and mutation rates. And of course, there's variation in fragmentation times within Pike Islands, because some of them are really high islands and some are low, and they actually didn't fragment it exactly 18,000 years ago. Some of them varied. And the finest resolution of the fluctuation rates is about 3,000 years. And we're getting close to that with our ability with genomic data to talk about the temporal framework for some of this diversification, and just the possibility for fine scale augmentation of the Pike diversification model, which embraces biodiversity at the finest scale at which it naturally occurs. So I'm pretty excited about that. We need to thank our sources of funding here, and I want to thank the University of Kansas for all the support over the years and grants to 3 of the people who are involved in all these publications that we talked about today. Again, if you ever get a chance to go to the Philippines, it's a great place, not just to go to be a tourist, but also to be a biogeographer, because the slogan is, you know, more fun in the Philippines, and it's more fun to do biogeography in the Philippines for sure. I really want to thank my lab group back at Kansas, which now involves finally a bunch of Filipinos who are focusing on a lot of this research. It's a big diverse group, and we have a lot of fun doing that stuff. Every year we do a band photo cover, you know, an album cover. So this is last year, and my colleague Mitch Glore in the background there. So yeah, thanks to all of them, and thanks to the people on the other side of the Pacific who do all the work in the field and acquire all these populations. This is my dream team, field team here, and I won't go into too much of that, but some of you all heard more about them on Monday night at Herp Group. But yeah, thank you again for the invitation. I'd be happy to answer any questions you have. Thanks.